Hello, my name is William Rigby, and I'm a professor of medicine at Dartmouth Medical School. Today we're going to talk about T-cell ontogeny and tolerance, and in particular the case of AIRE deficiency and how it informs our thinking about this process. My program today addresses Chapter 2 in the program of Basic and Clinical Immunology. Our books and resources are all available from Garland Science, and they involve The Immune System by Peter Parham and Immunobiology edited by Kenneth Murphy, and the relevant chapters are shown below. So our learning objectives for today are the following. We're going to describe the structural basis of recognition of self by T-cell receptors and the role of the MHC, major histocompatibility complex, in antigen presentation. Now, I want to remind everybody early on that the MHC is the same thing as HLA. The second point we're going to focus on is how does MHC restriction occur by the T-cell receptors, and how does this lead to differentiation of CD4 or CD8 cells? This process in toto is called positive selection. Third, we're going to recognize the pathways that delete autoreactive T cells, the so-called negative selection, that leads to central tolerance. Fourth, we're going to describe the overlapping pathways of tolerance, the peripheral versus central tolerance pathways. And we're also going to use the autoimmune regulator deficiency, AIRE deficiency, as an example. So, as we begin our conversation about T-cell ontogeny and tolerance, the central question we have to ask is how does our immune system, specifically our T cells, discriminate self from non-self? And we're going to do this in 4 bullets that have been largely captured in the learning objectives. And we're going to first talk about the structural basis of recognition of self by T-cell receptors and the role of the MHC in antigen presentation. When we talk about this, it's best to have an overview of the entire process of T-cell ontogeny. Shown in this slide are 4 panels. On the far left-hand panel is an immature T-cell that has just migrated to the thymus. And within the nucleus of this schematic, you see a T-cell receptor gene that is about to rearrange. These T-cell receptors are rearranging in a pre-T cell that migrates to the thymus from the liver, or the bone marrow. And then in the thymus, shown in the second panel, it rearranges its T-cell receptors such that it can bind to or not bind to self. The cell with the condensed nucleus in the upper left-hand, in the second panel in the upper left-hand corner, is an apoptotic T cell because it has too much recognition with self, and that needs to be selected or deleted. The second T cell has some reactivity with self, being able to bind and recognize the MHC, but it's not being deleted. Subsequently, these T cells are able to migrate to the periphery and function in the lymphoid organs, and finally, in the last panel on the right, mediate a effector immune response that protects us. So when we talk about this, we have to return to our central question, is how do T cells discriminate self from non-self? And we have several points we have to cover to get to this, to address this question. The first question is just what is self? And self is every protein, every carbohydrate, every fat molecule, every lipid molecule in our body. So these are proteins that we all share, whether they be cytokines, whether they be insulin, whether they be a cytoskeletal protein like vimentin. They're shared among all of us, all human beings, and they're identical among all of us. However, to be able to discriminate self from non-self also means we have to have proteins that we do not share. And these are the different HLA genes, which are so important for allograft recognition and organ transplantation. These genes differ from individual to individual and are shared among family members. These HLA genes define the differences between self or selves. Now there's HLA A, B, and C, and these are defined or generally referred to as class 1 MHC. We also have HLA-D, or DR, or DP, or DQ, which define class 2 MHC for all species. So the question about self and non-self returns to the immune system, and we have two basic mechanisms of recognizing pathogens or foreign invaders. One mechanism is the antibody, shown in the panel on the left in the upper left-hand corner. Antibodies do not discriminate between self and non-self. They just react with a particular protein or a particular structure. So therefore, we need to generate our ability to recognize self from non-self through a different pathway, and in this pathway, we use the T cell receptors, or the TCR. The TCR recognizes pieces of protein, so-called peptides. They're usually about 8 to 14 amino acids long. But they only recognize these peptides in the context of self, that is, in the context of the HLA genes that you have. This is the so-called process of MHC, major histocompatibility complex restriction. To avoid autoimmunity, these T cell receptors cannot react with normal host proteins. If they did, they would mobilize an immune response. So the big question is, how does this happen, and how does the thymus do this? It generates the ability to discriminate self from non-self, but also eliminates T cells that react with self too strongly. That is, autoreactivity. So this is shown in the following diagram, and I want to take you through it from left to right in the following manners. On the left-hand panel, we have a T cell receptor in the upper half of the panel responding to an antigen-presenting cell, an MHC molecule. And shown in this yellow MHC molecule is a peptide that we'll call X. And this is a T cell receptor that's specific for that peptide in the context of that MHC molecule. In the middle panel, there is that same antigen, that same peptide, peptide X, is being presented to the same T cell, but this time the antigen-presenting cell comes from a different individual, it's so-called MHC-B instead of MHC-A, shown in yellow. There's no recognition in this pathway, emphasizing that the T cell receptor is not seeing the peptide, such as might occur with an antibody, but it's seeing the peptide in the context of the MHC molecule, the correct MHC molecule. The third panel on the right emphasizes this particular point. Same T cell receptor, same MHC as shown on the far left-hand panel, but now we have a different peptide, and there's no recognition. So the T cell receptor sees peptide and MHC, but the only MHC it sees is the MHC in which it was raised by, that is, so to speak, in the thymus. So why do we need to recognize self? Well, this is pretty obvious, but let's review it anyhow. The first thing is that when we want to recognize self, we really want to tell the good guys from the bad guys, okay? So bad guys are shown in the right-hand panel. There are two kinds of pathogenic bad guys. One are the bacteria or fungi or whatever, the things that live extracellularly. The second type of pathogenic bad guy are the viruses, because they live inside cells, so they're not amenable to direct attack outside the cell. Therefore, two different pathways have evolved to discriminate these pathogenic bad guys from the self. The self is basically the molecules that are found all over your body, and also the molecules that are only found in certain places, like in your retina, like rhodopsin, or in the brain, like myelin basic protein. Very important that you have the flexibility and the ability to discriminate both universally expressed proteins as well as tissue-specific proteins. Now, as it turns out, we've evolved two different pathways for recognizing these bad guys. For intracellular proteins, we have CD8 positive T cells, and they recognize antigen presented by HLA A, B, or C. And we also have extracellular pathogens recognized by CD4 positive T cells, which present antigen in the context of HLA-D. Now, these two kinds of self are shown here in a diagrammatic on the left-hand side. In the upper panel are ribbon diagrams of the MHC class II and the MHC class I, silver representing the backbone in which those molecules exist. And orange in the left-hand panel shows the CD4 molecule from MHC class II actually binds to that silver backbone. And CD8 actually binds to the MHC HLA A, B, or C that's shown on the right-hand panel. Said differently, not only do your CD4 and CD8 molecules only are expressed on T cells that recognize class 2 and class 1 respectively, they also act as adhesion molecules which hold that complex together. So the question is, how does this happen and why does this happen? So for this, we got to take a step back and do a little brief review of antigen presentation. And we're going to focus on extracellular antigen presentation that CD4 positive T cells and HLA-DR molecules participate in. And I just show here on the left-hand panel is an extracellular antigen that's being taken up by an antigen-presenting cell. It gets put into an endosome. The pH drops, shown in the third panel from the left. And as the pH drops, the proteases that are in that endosome become active and start chewing that protein up into little red blobs. A second vesicle fuses with that vesicle, and that second vesicle has HLA-DR or D molecules, and it picks up that peptide and moves it out to the cell surface. Once it's moved out to the cell surface, you see the panel, in this diagrammatic model, it's moved out to the cell surface where it's presented to CD4 positive T cells, shown in the far right-hand panel. B cells do this, macrophages do this, dendritic cells do this. Now the left-hand panel also describes the other kind of recognition of self that occurs with cytosolic or intracellular pathogens. And this is any cell in the body. Any cell in the body can be affected by a virus. These viral particles are being made in the virus inside the cell as it tries to replicate. Proteins are generated, and some of those proteins get degraded and expressed on the cell surface bound to MHC class 1, or HLA A, B, or C if you happen to be a human. CD8 positive T cells then that are specific for that viral protein in the context of that HLA A, B, or C molecule lead to the development of an effective antiviral response. Now, this is a reminder of just what the T cell is actually seeing. Shown in this panel are a ribbon diagram, sort of in silver at the top, and a ribbon diagram with silver at the bottom. And this represents the MHC molecule, whether it's HLA A, B, or C, or DR. The linear portion with multicolor with blue and red dots lying in the middle is the peptide. The peptide can be generated from a virus, in which case you knew that this would be from an HLA A, B, or C molecule, or it can come from an extracellular pathogen, in which case it would probably be an HLA-DR molecule. And these are presented to T cells. But the T cell, remember, is seeing the upper half of the molecule, the lower half of the molecule, and the peptide. And if that peptide is foreign, it mobilizes an immune response. If that peptide is a self-protein, you do not want to attack that protein because that would cause autoreactivity and possibly tissue destruction. So the second question is, why do we need to recognize self? Well, the mission is we need to recognize self. That's what we call MHC restriction. The second mission is we have to protect against invaders and get the host to a reproductive age so they can pass these genes along. And the third role that seems to be more and more important in our period in adults is that of avoiding autoimmunity, and this is called tolerance. This process occurs in the thymus. This T cell education, or this process of T cell ontogeny, occurs in the thymus. And it occurs in the two different compartments of the thymus, the thymic cortex, which is where MHC restriction, as well as commitment to CD4 or CD8 positive T cells occurs, as well as in the medulla, which is where deletion of autoreactive T cell receptors occurs. Shown here is the schema of the thymus. And what I want you to notice in the panel to the right, the photomicrograph, is that the cortex is very, very cellular. And it's actually more cellular at the top and less cellular as you move down to the bottom. That's because T cells are being killed off. And then finally, you see below that area, and the least cellular is the area of the medulla. This is shown in a diagrammatic model on the left-hand side. And what I want to point out to you is those dark blue cells are all lymphocytes, and those lymphocytes are sort of percolating through the thymus and bumping into cortical epithelial cells. And after they bump through, they sort of percolate through that area, they then move into the medulla where they're going to interact with dendritic cells as well as medullary epithelial cells. And these are all shown on the diagram, and we'll return to this in a second. So, in the cortex is where MHC restriction and single positive T cell generation occurs. When T cells get to the thymus, they start rearranging their T cell receptors. As they do so, they express both CD4 and CD8 on their cell surface. They are so-called double positives. As they percolate through the thymus, they are destined to die. 99% of those T cells die. They die if they don't generate the right kind of T cell receptor. Their T cell receptor in the thymus has to bind the MHC in the context of some self-proteins or self-peptides that are bound to the MHC molecule that are present on the cortical epithelial cells. Now you can, since the T cell receptors are being randomly generated through recombination and so forth, you can imagine that there's a lot of mistakes. And the mistakes will be in both in the alpha chain and the beta chain that make up the T cell receptor. And without both of them being productive and able to recognize self, they just kick. Now what's interesting is that the T cell receptor that cannot bind the MHC, they die. So thus what you've generated are T cell receptors on your T cells that can bind self-MHC, that is MHC restriction. Now the T cell receptors that bind to class 1 or HLA-A, B, or C T cell receptors differentiate to CD8 positive T cells, and they get rid of CD4. The CD4 positive T cells are generated from double positive T cells whose T cell receptor recognizes class 2 or binds to class 2 a little bit better. So as a result, you get not only MHC restriction, you also, that is, the T cell receptor only sees class 1 or class 2 MHC and only sees self-MHC, but you also get your T cells differentiating to a CD4 or CD8 positive T cell. So we see this process shown here in this arrow, and what I want to emphasize is the T cell receptor binding, or to HLA, determines the commitment of the CD4, CD8 double positive cell to CD4 or CD8. And this leads to CD4 positive T cell receptors recognizing HLA-DR, and CD8 positive T cells recognizing HLA-A, B, or C, which make up the class 1 MHC panel. Now, the whole question of central tolerance comes from the medullary thymus, medullary portion of the thymus. And this is where you've already selected for T cell receptors that bind to self-MHC. And that you've done that in the cortex, but you don't want them to bind too tightly or react too strongly. You just want them to have low affinity interactions. And so what you have to do is you have to get rid of the T cell receptors that bind MHC self-peptide and the too strongly. And the way that occurs is when those T cells bump into a medullary dendritic cell. So in contrast to the cortex of the thymus, where T cells have to be rescued from cell death by having the T cell receptor that engages MHC, in the thymus, if your T cell, in the medulla, if your T cell receptor engages the MHC too well in the medulla, when it interacts with the dendritic cell, it dies. And this is the process of deletion of the lack of autoreactive T cells to proteins that are expressed in the thymus. So this is how we generate tolerance to all the proteins that are expressed all over our body. But the big question is, how do you generate tolerance to proteins that are not expressed in the thymus? Like some proteins that are like in the ovary, or in the sperm, or in the brain, or in the eye. And a fascinating second pathway exists, and I'm going to tell you a little bit about that right now. So this model is described up here when we talk about the autoimmune regulatory gene. Shown in the left-hand panel, we have the individual organs of the body express tissue-specific antigens. So we have retina proteins in red, and we have ovarian proteins in blue. You can measure pancreatic proteins in yellow, and so forth and so forth. In the thymus, the T cells arise that are shown in those colors that recognize those particular proteins. T cell receptors arise, they are capable of recognizing specific antigens. And what happens in the medulla is that there's a specialized cell which, under the control of a specialized protein, actually expresses those proteins. And that's the role of the AIRE gene, A-I-R-E, the autoimmune regulatory gene. And the fascinating thing is that medullary epithelial cells express rhodopsin, they express insulin, they express various different proteins that you would never think would need to be expressed in the thymus. And their whole point of being expressed there is to delete autoreactive T cells to these tissue-specific proteins. And that's shown in the third panel, that you see these T cell receptors that are binding too well to those cortical epithelial cells that are expressing those peptides that they're autoreactive with, and what happens is those T cells die and never escape the thymus. In contrast, if you have AIRE, if you have a deficiency in AIRE, which is a recessive gene, and it's found in certain populations of the world, such as in Finland, the T cells do not get deleted. And what happens is that these T cells with T cell reactivity against self proteins persist in the circulation and actually cause an autoimmune disease that I'm going to talk about shortly. So as we talk about this process, I emphasize this whole point in this last slide, that the AIRE in the medullary epithelial cells allows the deletion of T cell receptor reactive with tissue-specific self-peptides. And that's very different from the negative selection of widely expressed proteins and peptides that occurs on the medullary dendritic cell. So there's two different cells doing two different kinds of negative selection in the generation of central tolerance, which means the deletion of autoreactive T cell receptors. So let's review this. We start off with the double positive T cell, and in the thymus, these T cells become restricted to a particular HLA and to learn how to recognize self. That's MHC restriction. The second process is the commitment of the CD4 or CD8 cell from a double positive precursor. And finally, central tolerance is generated by the deletion of autoreactive T cell receptors with either general self-proteins or with tissue-specific self-proteins, insulin and so forth. So it's clear that central tolerance is imperfect. So many other forms of tolerance exist. You can imagine that you can't select for every protein in the body when there's so many different proteins. And what's interesting is how evolution has evolved with the AIRE gene to select against particular proteins, but obviously it doesn't select against everything. So we have the persistence of autoreactive T cells. So how do we control those? Well, there's 4 or 5 basic mechanisms. One, we have the central tolerance, which you talked about at the top. That's deletion or editing, and that occurs in the thymus or bone marrow for both B cells and T cells. We're going to focus on T cells. Antigen segregation. Antigens, like in the brain, are kept physically distinct away from the immune system. Peripheral anergy, cellular inactivation by weak stimuli. That is, if an antigen is presented to a T cell without a good co-stimulatory interaction between CD28 and B71 and B72, CD80 or 86, you don't get an immune response. We have regulatory T cells, which actually actively suppress this immune response, and regulatory T cells arise by a different mechanism in the thymus. And then we have functional deviation, meaning there's some immune responses that if they're more related to a Th2 response, they don't cause any tissue injury, as opposed to if they're a Th1 response, where they cause a lot of tissue injury. And activation-induced cell death, of course, is when a T cell encounters an antigen and starts to react with it when it shouldn't and undergoes apoptosis. So there's many different pathways of tolerance. And we're going to talk a little bit about these when we talk about what happens in APECED. So what happens if in this pathway, if AIRE deficiency does not work, AIRE deficiency is present, if these elements don't work? So we're going to talk about APECED, which stands for Autoimmune Polyendocrinopathy, Candidiasis, Ectodermal Dystrophy, which is a mouthful. And I'm not going to talk a lot about the ectodermal dystrophy, but I want to show you that in a Finnish cohort, the patients with APECED had this. They had hypoparathyroidism, so they had an immune response directed against PTH. They had adrenal failure. They had an immune response directed against their adrenal glands. Ovarian failure, 60%. Interestingly, diabetes, that is an immune response directed against the islet cells and insulin, only occurred in about 18%. So you can imagine all these represent partial failures of tolerance. And yet we know that APECED has not selected for, has allowed autoreactive T cells to persist in all of these. If central tolerance was the only mechanism by which we protected ourselves against autoreactivity, you would expect hypoparathyroidism, adrenal failure, and ovarian failure, and insulin-dependent diabetes mellitus all to be 100%, but they're not. So clearly there are other pathways of tolerance that we alluded to. Now the exact balance and contribution of these factors is not certain. Now, one of the most fascinating things about APECED is this business about candidiasis. Now, candidiasis is mucocutaneous candidiasis. It's present in 100%. Why is that? That is so crazy. When you think about this, you have an immune deficiency that's actually associated with increased sensitivity to a particular fungal infection or yeast infection in this particular case. And the reason is because you develop an autoimmune response against IL-17. So isn't this fascinating? You have autoantibodies that arise in these patients due to the failure of negative selection. They're actually directed against other T cell products. And the other point that this really makes, and this is only partially understood, is that what this really defines is how central IL-17 is to the need for host immunity to Candida. This is a fascinating observation, and there's many things that need to still be figured out, but it really establishes that everybody with APECED makes autoantibodies against IL-17, and that predisposes them to increased sensitivity to Candida infections. Fascinating, and an area I'm looking forward to following over the next decade or so. So as we come to our conclusion slide, I want to emphasize that we recognize self through T cells that express receptors, TCR, that recognize the peptide fragments, but only in the context of self. And that ribbon diagram I showed you with the peptide in the middle is true for HLA, A, B, C, or D. This is the concept of MHC restriction. There are two specialized T cell pathways. There's CD4 positive T cells that recognize peptides presented by HLA-D proteins or D antigens. CD8 T cells recognize peptides expressed by HLA-A, B, or C proteins, which are found on every nucleated cell in the body. Finally, the MHC restriction and the commitment to CD4 or CD8 lineages are determined by positive selection of T cell receptors that react with these peptide complexes expressed by cells in the thymic cortex. So there's a clear partitioning of this to the thymic cortex. However, central tolerance occurs when we eliminate T cells in the thymic medulla, where T cells expressing T cell receptors that recognize self-peptides too well are eliminated. This deletion of autoreactive T cell receptors to both general and specific proteins occurs in the medulla, but through two different cellular pathways. One is the dendritic cell, and one is the medullary epithelial cell, which expresses AIRE. Now, there's other pathways of tolerance that provide peripheral tolerance, many of which, in fact, most of which, are functionally redundant to the concept of central tolerance. So the restricted clinical manifestations of the AIRE deficiency, the APECED syndrome, demonstrate the key role of central tolerance in preventing certain forms of autoimmunity, but at the same time establishes that that cannot be the whole story. Otherwise, you would see autoimmunity to all endocrine hormones, which we do not see. So with this in mind, I'd like to thank you very much for being able to share with you this fascinating biology, and in subsequent talks, we will talk about its relevance to various components of the immune response and disease. Thank you.